There are two important procedures, or you can say the important activities related to the analytical procedures, and those are the verification and the validation. For the product, or you can say the pharmaceutical product, we refer the method available in the pharmacopoeia, or if it is not there, then we need to develop the method. This video is related to the verification of compendial procedures. Wherever the procedures are developed or these are used from the pharmacopoeias, these are required to be verified. When developed in-house, these are required to be validated. I welcome all the viewers to this video on the USP chapter number 1226, verification of compendial procedures. All the USP chapters are very much important for us, and we should know the chapters and how these are related to our daily work. So this chapter covers the verification of compendial procedure. It explains how to verify a compendial test when using it for the first time. This chapter ensures the method works with your lab personnel, equipment and reagents, and it is not meant for already established successfully used methods. Then two terminologies are there, mainly verification and validation. Validation is full analytical evaluation of the new methods, and USP chapter 1225 covers it. Verification is for checking the selected performance characteristics only for the method, and it is covered in the USP chapter number 1226. The aim or objective or goal of this chapter is to confirm that an official USP method works as intended in your lab. Many of the times the minor variations can lead to the problems, can lead to the results which are not intended or which are not expected. So verification is required. Now we will go through the regulatory requirement. We will check the regulatory requirements. Labs do not need to validate the USP methods because these are already developed methods, and we need to check those methods whether these are applicable to us or what are the challenges involved, what we can do to use those methods, because these are ready methods for your use. So labs do not need to validate the USP methods, but they must verify suitability under the actual use. Based on 21 CFR, suitability of all testing methods shall be verified. If your formulation is enlisted in the USP, if method is available in the USP, so you can take that method and verify it. Now, what this chapter does not cover. So, verification chapter is not related or cover the microbiological procedure verification. It is excluded because it is covered in the other chapters. So, chapter number 51, 56, 51, 61, 62, 71, 1 to 27 is for the microbiological procedure verification. 51 number is for microbial effectiveness, 61 is for microbial enumeration, 51 number chapter is for antimicrobial effectiveness, 62 number test for specified microorganisms, 71 for sterility test, 1227 for microbial recovery validation. Then verification process. So we should understand the compendial method, have required training and experience, perform verification to ensure the method works with their sample. If verification fails, you can consult USP staff, you can communicate. If still unresolved, method may not be suitable. Develop and validate an alternative method as per the requirements, as per the principles and procedures. Then when to verify? So verification depends on the method complexity, type of sample, whether it is a drug substance or product, instrumentation, user experience and training, nature of excipients or impurities. Only relevant performance characteristics need the evaluation. So this is the information points for verification process and when to verify. Now coming to the key parameters for verification, because verification is not a validation. So it is a process of verification. Depending on the method, you can evaluate the specificity, LOD, LOQ for impurities, precision, system suitability. System suitability like resolution of chromatography. Not all parameters are required, only the ones appropriate for your article and the method are required to be verified. Example, why specificity matters. So different API suppliers, different impurity profiles will be there. It is not the same ROS followed by the different manufacturers. Different manufacturers will have different chemical reagents, starting materials they are using. So impurity profile will be different. That's why specificity matters a lot. Different excipients in the products will give you different interference. Different degradation products may be formed because of the different excipients. Buffers, antioxidants and extractables can affect the results, and therefore specificity must be confirmed for your particular sample. When verification is not required, so this is also very important point. You should know when verification is not required. Not needed for the basic routine compendial test like loss on drying, residue on ignition, acid value, pH measurement, or simple wet chemistry tests. Exceptions: if using the method on a new article, check if the sample preparation or handling is different. So verification is proving a USP method works in your lab for your sample, and verification is not a full validation. It is to evaluate the relevant parameters, and it checks whether the method works for your product or you in your lab or not. So the documentation required as per the GMP requirements, and whenever you have the compendial method, always do the verification. Always check the method with your lab instruments, with your analyst, with your procedures, with your SOPs, and then you can use it. So this was the information for verification part. In the upcoming time, I will be making more elaborative videos on the USP chapter. So uh, stay tuned to this channel. Share the videos with your friends and colleagues. And thank you for watching the video. Let's meet soon with another informative video.